Welcome to APBiology 3.1 to 3.3, Metabolism and Enzymes. So, in unit number 3 is all about cellular energetics. The first 3 parts of this unit are about enzymes. And after that, we discuss cellular energy and then we focus on photosynthesis and cellular respiration and finally fitness. Now, this particular conversation is going to focus on units 3.1 to 3.3, chapters 3.1 to 3.3, and a little bit of conversation on 3.4 as well. This is going to be quite a long chapter, but it lays a solid foundation for the rest of our conversation on photosynthesis and cellular respiration. So as you can see here, there are multiple learning objectives, and this in its entirety covers all the key things you need to know about enzymes. So we're going to start with how biomolecules keep and use energy. And then we talk about endergonic and exergonic reactions, catabolic and anabolic processes, and how energy is coupled. Then we look at energy graphs and what free energy means. Then we look at enzyme structure and properties, and what an active site means, and what is activation energy. And then we look at the properties of enzymes. After that, we look at the mechanisms of enzyme action. There are two main theories or models for enzyme action: lock and key model and induced fit model. Then we look at competitive and non-competitive inhibition, how enzymes are inhibited, and then how enzymes are activated. So cofactors and coenzymes and cooperativity. Then we look at the factors, the environmental factors that affect enzyme activity. That includes pH, temperature, substrate concentrations, enzyme concentration, salinity. And then we will look at how enzymes tend to work best in optimal conditions. And when you exceed a certain limit, then the enzymes denature, and in some cases renature, reform. And finally, a very quick note on how enzymes are named. So if this is a very long list and you find it daunting, I can assure you that it all ties into each other. It all makes sense. It all comes together very nicely. But if you do need to stop and break it down into multiple chapters, please do so. All right, it all starts with the sun. Everything starts with the sun. The nuclear fusion reactions in the energy of the sun, in the core of the sun, gives rise to the intense amount of energy that travels this great distance, great void of space, finally reaches Earth, and it meets the plant life, the chloroplast. Inside the chloroplast, the magic of life happens, where the energy of the sun is captured into the form of chemical bonds. And these chemical bonds, now captured in organic molecules, carbon, hydrogen, oxygen, are used by the plants and by the animals that consume them in mitochondria. Mitochondria, cellular, is where cellular respiration happens. And simply what cellular respiration is that the chemical bonds that are contained in these molecules of carbohydrates that the plants have created are broken, and the energy is released and then transformed and not transferred, transferred into small molecules that can carry the energy all over, which we call as the energy currency of the cell. These are ATP molecules, adenosine triphosphate molecules. So in other words, what has happened is the energy of the sun is now captured in the chemical bonds between atoms, and whether they are present in the larger molecules or smaller molecules, it is all there. In the energy of the sun is now in the chemical bonds. And so as these bonds break, the energy is released. And as new bonds need to be formed, this energy is used. That in a nutshell is the process of how energy is carried in the form of life. So we need to particularly look at the ATP molecule. We looked at nucleotides when we were studying DNA, right? In our introduction to DNA. DNA has 4 nucleotides, A, T, G, and C. The A is for the adenosine. So if you look at it quite closely, so the nucleoside part of a nucleotide is the part that contains just the sugar and the nitrogenous base. The sugar and the nitrogenous base is called as the nucleoside. The sugar and the nitrogenous base and the phosphate together is what we call as the nucleotide. A, T, G, and C are all nucleotides. Adenosine is a nucleoside, meaning we are only talking about the A nitrogenous base and the sugar part of it. And if we have one phosphate group attached to it, that nucleotide is called as the adenosine monophosphate. Now, when energy is released by the breaking down of these carbohydrates, some of that energy is now moved to form a chemical bond between a phosphate that comes swimming down and the adenosine monophosphate. So you see that energy from breaking down of carbohydrates is now captured right here. At this point, adenosine with the 2 phosphates is called as adenosine diphosphate or ADP. You can see that here. So you have the nitrogenous base, the ribose sugar, and the single phosphate together is the adenosine monophosphate. And some of the energy from breakdown of carbohydrates comes and is used for making the additional phosphate bond. So now the energy is contained here in this little red line you see here. When more energy comes in, then another phosphate is brought in. And now you have some more energy captured in this molecule. So adenosine triphosphate, meaning 3 phosphates. Now you have energy stored between the first and the second phosphate groups and the second and the third phosphate groups. When energy is needed, these phosphate bonds are broken. All of these happen because of enzymes. And the enzymes come and they break down these phosphates and release that phosphate group to go out. But when the bond breaks, the energy is released, and that energy can be used for constructing other biomolecules, for other properties, for other activities of life. Okay. So, when we look at the kinds of chemical reactions in a system, they are either anabolic reactions or catabolic reactions. Anabolic reactions are those that are helped in constructing larger molecules, making larger molecules. And catabolic reactions involve breaking down of large molecules into smaller molecules. So think of ants. Ants build things. Cats like to break things. That's how I remember. Anabolism, building stuff. Catabolism, breaking stuff. So you have smaller molecules, and these molecules need to come together to form larger molecules, meaning that they have to form chemical bonds between them, which means they need energy to form chemical bonds. Okay, so anabolism requires input of energy, whereas catabolism means you are taking a large molecule and breaking it down into smaller molecules, which means you are breaking chemical bonds. And remember that energy is held inside chemical bonds, and that means when the bonds are broken, energy is going to be released. Now, when we eat stuff, okay, whatever food that we eat, whether we are eating proteins, carbohydrates, or fats, they need to be broken down and then reconstructed in whichever forms our body needs it. So, for example, proteins are made of amino acids, you know that. And when amino acids are broken, energy is going to be released. And that energy is going to be used for making ATP molecules. Same thing here. You have carbohydrates. Carbohydrates are broken down, and the energy is used for making ATP molecules. And then looking at fats, once again, you're using the energy contained in the bonds between carbon, hydrogen, the fatty acids, and glycerol. So that again leads to release of energy. So the energy is stored in ATP molecules, and again ATP molecules are broken down when the body needs to make stuff to synthesize, for example, new proteins. Then you're going to need some ATP molecules broken down into ADP, and that energy released is going to be used for constructing new proteins. So speaking of breaking down things and building things, we saw this before. That is how biomolecules are built. We saw that whether we are looking at nucleic acids or proteins or carbohydrates, it involves polymerization of monomers. And when things are brought together to create polymers, what you're doing is you're building chemical bonds. This is dehydration synthesis, where you're losing water so that you can form a covalent bond here. This dehydration synthesis and formation of a chemical bond is going to require energy. Okay, so this is going to be an anabolic reaction. Here you have hydrolysis, when you have a polymer being broken down to make monomers, and water comes in, and the water molecule is used to break this complex polymer into smaller pieces, and that's why it's called hydrolysis, using water to break stuff. And because chemical bonds are broken, energy is going to be released. In biological systems, this breaking down and constructing is linked to each other, because to create something, to create a polymer from a monomer, you're going to need energy. And where do you get that energy from? Usually from ATP. So ATP, you saw the food that we eat has energy stored in the chemical bonds, and that is converted into ready-to-use molecules called ATP. And the ATP has energy stored in those little bonds between phosphate and phosphate, phosphate and phosphate here. And so using enzymes, the ATP is broken down to ADP and PI. So when that bond is broken, some energy is given out, right? That is the energy that is driving polymerization. So the breaking down to release and releasing energy is linked to building by using energy. This is called coupling of two biological reactions. Energy of one is used to create the second reaction. So here's an example of coupling. So we have glucose being broken down, okay? And this is just what I explained. Glucose being broken down, and carbon dioxide and water and heat are released. And the energy that is created from here is used to convert ADP into ATP by taking an inorganic phosphate and then adding that energy to make ATP. Okay, so now this energy is stored. Energy is potential. The energy stored here is the potential energy stored in the form of, ready to use whenever you need it. And then when the need comes, amino acids are going to be linked together to form proteins using dehydration synthesis. And for that, this energy is going to be used by breaking down this. So in the process, then you're going to release ATP. So that's an example of coupling of energetic reactions. So here I want to introduce two words, Endergonic and Exergonic. So we saw here that glucose is broken down, right? And energy is being released in the process. Whenever you have a reaction that releases energy, that's called as exergonic. Okay, exergonic. And if the conditions are right, the energy is released spontaneously. The reaction occurs spontaneously, happens very quickly. And we saw here that there is a situation where you have to use energy to make something else. So when you have an input of energy to drive a chemical reaction, it's called as endergonic reaction. Okay, so you need to remember the difference between endergonic and exergonic. So just take a look at the pictures here, because you're going to discuss this in a little bit more detail. So we can represent all of the energy play between molecules in the form of energy graphs. So here we are tracking the progress of the reaction from reactants to products on the x-axis. And then we are looking at the total stored energy, potential energy of the molecules. Okay, so you look at the reactants here. Here the reactants have a high level of energy compared to the products. So under the right conditions, the reactants are just going to spontaneously break. Because when you have a lot of energy, the natural tendency is to give away that energy. So the energy is released. Okay, so this is an exergonic reaction when you're releasing energy. On the other hand, you have reactants that start off with a lower level of energy, and your products, the products that you want to create, need are meant to have a higher level of stored energy, higher potential energy, which means all of that energy has to come from outside. That is the energy input. Therefore, this is the endergonic reaction. So now look at the same thing in dehydration synthesis and hydrolysis. Dehydration synthesis is going to need energy input. So would that be an endergonic or an exergonic reaction? It is going to be an endergonic reaction. Same way, when you have hydrolysis, where you have release of energy, that's going to be an exergonic reaction. So look at this. This is going to be exergonic. This is going to be endergonic. So there's one more term I want to introduce. It's the activation energy, otherwise represented as EA. Okay. So it takes, just as we saw, where was the one? So for running a chemical reaction generally, okay, so you're going to need an application of energy here. So you're going to need an application of energy. So let's say I'm drawing an energy graph, and I'm looking at how much time it is taking for a substrate to become a product. Substrate is the starting material, reactant, okay, and the end product is here. And then you're looking at the amount of energy on the y-axis. You need to keep applying energy to a substrate until it comes to a transition point where the chemical bonds are broken, and then a downstream reaction starts. The reaction downstream is usually much faster, but here you have to keep applying energy. But when you have a catalyst is something that can move the reaction forward much more quickly, with much less energy. Okay, so when a catalyst is present, what happens is the amount of energy that you need to kickstart the reaction to get to this transition point is reduced dramatically. Okay, so that is essentially what enzymes are. Enzymes are biological catalysts. What they do is they reduce the amount of activation energy that is needed to kickstart the reaction. Enzymes tend to be much larger than their substrates. So, for example, catalase is an enzyme that works on hydrogen peroxide, converts it into water, and releases oxygen. And there are, the possibility of hydrogen peroxide automatically breaking down into water is much less. But when you have the catalase, it can catalyze or make reactions happen at the rate of millions of hydrogen peroxide molecules processed every minute. And if you see again, because the enzyme is so small, so big compared to the substrate, there is a small portion within that enzyme where all of this magic happens, where all the binding to the substrate happens and where the catalysis happens. That region on the enzyme is called as the active site. So if we think of an active site is, let's say we have an enzyme that is about 500 amino acids big. Okay, just two of those nucleotides, two of those amino acids are essential for actual catalysis, and probably 2 or 3 more are required for binding. So you can see how small an active site is compared to the size of the enzyme. So there's a small place within the enzyme where the binding to the substrate happens, and a couple more amino acids that help do the catalysis reaction. It's easy to visualize it in this form though. So you can see the enzyme coming to the substrates coming to the enzyme's active site, binding in there, and because of the close proximity and orientation and all of that, the reaction is able to go much faster, and then you form the product. But the beautiful thing is the enzyme is not consumed in the process. After the product is formed and it goes out, it comes back to its original shape. Okay. So what I want to also introduce here is the idea of free energy. Okay. So let's say this is the starting energy point. Okay. And then this is the ending energy point. In this example, we have the substrates having more contained energy than the products. Of course, you're going to take a bit of energy to go all the way to the transition point. And then very, very quickly, you're going to have a downstream reaction. Products are going to form very quickly. There is a point here though, as the substrate are formed, when the substrate, when the product is being formed, where the energy of the product being formed is the same as the substrate. So anything else after that is energy that is going to be released. Extra energy, right? Energy is going to be released out. So this energy that is released, that is the difference between the substrate's energy level and the product's energy level, is called as the free energy. It's written as delta G. Okay, delta G. That's the free energy. It's otherwise called as Gibbs free energy. When you have in the same situation, you have an enzyme, and the enzyme is able to reduce the need for activation energy, how much energy it needs to kickstart the reaction, but still the product that is formed has the same lower level of energy as would happen without the enzyme. So that's not going to be affected. So you still have the same net energy that is released. In other words, delta G, or the free energy, is not going to be affected by the presence or absence of enzymes. What enzymes are actually doing is they are reducing the energy needed for activating the chemical reaction. So here you have an example of lactose. Lactose is a disaccharide made of glucose and galactose, two monosaccharides. They are held together by a bond. Now lactose comes, and if you just wait for lactose, if you just have lactose sugar, and you wait for a long period of time, just the amount of energy for it to spontaneously form glucose and galactose, to break down into glucose and galactose, it's going to take a really, really long time, and it's going to take a lot more activation energy to do that. But when you have the enzyme lactase, lactase has an active site for binding of lactose, and lactose comes and binds to it, and the binding in that small active site region puts a strain on the bond between the glucose and the galactose, and that causes the glucose and the galactose to break. So you needed much less energy because of the topological pressure applied by the lactase on the lactose bonds. And therefore, with much less activation energy, you are able to break lactose into glucose and galactose. Okay, so this is just a representation of the same thing. Okay, whatever I just described with the reactant, they are becoming the product. Without an enzyme, it's going to take a lot of activation energy. With the enzyme, it takes much less activation energy. So if you're given an image like this, you should be able to, and they say that this is a reaction without the enzyme, this is the reaction with the enzyme, you should be able to point out that this is the activation energy without the enzyme. So that's the line from the starting point, which is the substrate's energy, to the transition point. Okay, so that's for the enzyme, without the enzyme, and this is the activation energy with the enzyme. And this difference between the substrate and the product's energy is the free energy, otherwise represented as delta G. So how do the enzymes lower the activation energy in the active site? So we saw this example here where it was putting a topological pressure on the bonds. Again, when you have two substrates coming together, let's say A and B coming together to form a product which is called AB. Okay, what this enzyme active site is doing is it's holding the two substrates in very close proximity, aligning the bonds and the atoms of one molecule with the bonds and the atoms of another molecule. And because of the proximity, bonds can form very quickly than they would otherwise. And also, this active site can also create a local environment which is acidic or polar or non-polar for the reaction to occur. And so between the local environment and the proximity and the physical the topological strain that it is putting on the substrates, the reaction happens much quickly. And when as the substrate, the product is released, the enzyme reverts back to its original position. So the enzyme is going to be recycled. Which leads us to the properties of enzymes. Most of the enzymes are made of proteins. There are RNA enzymes as well, and they are called as ribozymes. Only a small amount of enzyme is needed because one, the enzymes are really large and they can process a lot of reactants at the same time. And also because enzymes are going to be recycled. They are going to be unchanged at the end of the reaction and used for the next process. And also enzymes are very specific. only each enzyme can only work on one chemical reaction. Catalase is going to work on hydrogen peroxide. Amylase is going to work on the carbohydrates in your it's present in the saliva, so it's going to work on the carbohydrates that you're trying to eat. And so each one of those enzymes has a specific function, specific chemical reaction that it can catalyze. Enzymes are affected by the environmental conditions, temperature, pH, salinity, the concentration of enzymes, concentration of substrates. And they need optimal conditions for them to work at their best. Optimal meaning ideal conditions for them to work at their best. So now we are going to start looking at the models of enzyme action. How do the enzyme bind to the substrate and make all this happen? The early stage model was this idea called lock and key. And what they thought was that enzymes have these perfect spots on their active the active sites, the active sites are perfectly designed to fit into the substrate. So when an enzyme encounters a substrate which is going to fit into it like a key fits into a lock, then it forms an enzyme substrate complex. And once that happens, then all the proximity situations and the pressure that we spoke about, all of these things will happen and make the enzymes happen quickly. But later on, it is realized that it is not always an ideal fit. The substrate comes and binds to the enzyme's active site, which is more or less a fit. But once it does that, then the enzyme's active site is induced or modified so that it becomes a perfect fit for the substrate, therefore forming the enzyme substrate complex. Because there is a induction of change, there is a modification in the active site of the enzyme by the binding of the substrate, we call it as the induced fit model. So here's the enzyme forming a complex with the substrate. But sometimes you have chemicals or proteins that actually block enzymes. We use a lot of this in our drugs as well, in our medical drugs as well, as a way to stop certain enzymes. So that process of stopping an enzyme forming a complex with its substrate and therefore blocking enzyme activity is called as enzyme inhibition. Enzyme inhibition, okay? There are two types of enzyme inhibition. One is called competitive inhibition. The other is called non-competitive inhibition. Again, just using the names, you can imagine what these two things are, two processes are. So here you have an enzyme, and this is the active site, and this is the substrate that is meant to bind to it, okay? But you have another protein or another molecule that can compete with the substrate to bind to this active site. And therefore, this kind of competition is called as competition to inhibit the activity of the enzyme is called as competitive inhibition. And on the other hand, the inhibitor can bind at a spot different from the active site. Okay, so the same enzyme, but there is a different spot on the enzyme, which we call as the allosteric site. Okay, an allosteric site is a different part of the enzyme where this competitor is going to come and bind. And as it does that, it's going to create a change in the enzyme's active site. So it's binding somewhere else, but the active site's shape is changing. And because of that, the substrate is no longer able to bind to it. So it's not competing directly with the active site, but it is changing the active site by binding to an allosteric site. This is called as non-competitive inhibition. And sometimes the binding of this inhibitor can be permanent. Okay, so it's called a suicide inhibitor. So you have an enzyme, and usually when you have an inhibitor, that competes for the space, it binds and it goes away. Um, or even when it comes and binds to the allosteric site, it binds and it goes away. But some inhibitors are designed so that when they come and bind, they bind irreversibly. And therefore, that enzyme is no longer active, permanently inhibited. So these inhibitors are called as suicide inhibitors, and the substrate cannot move them away. So this is called irreversible enzyme inhibition. And this is one more concept that we are going to look at more carefully in the future chapters. How do enzymes regulate their activity by themselves, right? How are enzymes' activity is regulated? So one of the concepts that we are going to study because of how important it is in our in several biological processes is called feedback inhibition. So here you have a series of reactions, okay, series of enzymes, because multiple enzymes, remember, are going to have their own unique functions. So you have an initial substrate. This enzyme converts it into a product which becomes a substrate for the next enzyme, that creates a product which becomes a substrate for the next enzyme, and finally you have an end product. It can be a very long chain of enzymes: enzyme 1, enzyme 2, enzyme 3, and so on. Okay, when you have enough of the end product and you need to stop this factory production, of materials, what you do is you want to stop the first enzyme or second enzyme or whichever enzyme it is from, you're basically putting a turn switch off in the manufacturing process, right? So what you do is, in many cases, what happens is the end product of the final of this final enzyme, this end product is going to go back and bind to an allosteric site on one of the earlier enzymes. So here it is binding to the first enzyme. So what that has done is, you can see the active site is now closed. So that way, the initial substrate is not able to go and bind into that space, into that active site. So now you have a product which has gone back, feeding back into the loop, and then inhibiting the enzyme sequence. This is called feedback inhibition. Again, we'll see real life examples of this in our future chapters. So that was inhibition, where you had an enzyme come in and inhibit the activity of an enzyme. How about activating an enzyme? Okay, so this is a normal enzyme. Okay, this is just a wild representation here, a cartoon. So you see here, enzyme binding to the substrates A and B so that it can make a product A, B, whatever. There is an allosteric site here. So an activator comes and binds to this allosteric site, the site away from the active site. Okay, that's the allosteric site. So the enzyme activator comes and binds to this site, and that changes the conformation of this enzyme's active site so that it can work with the substrates much more effectively. And so those activators that bind to this enzyme to make the enzyme even more active is called as the it can be of two types. They can be cofactors or coenzymes. So cofactors are those small inorganic compounds, ions, non-protein, like magnesium, potassium, iron, copper, zinc. So these can come and bind to this allosteric site and change the configuration of the active site so that it can attach to the substrates better. But when the activator is a organic compound, um, and which binds to the allosteric site. Then it's called as a coenzyme. Okay, coenzymes are also non-protein molecules, but they are organic compounds, and they are much more complex than the cofactors. Like you have riboflavin, which is vitamin B2, niacin, which is vitamin B3, coenzyme A, all of those things. Okay, so you should be able to differentiate between inhibitors and activators, and the name says it all. And then you have to differentiate between cofactors and coenzymes. Coenzymes, organic; cofactors, small inorganic. So here's blood. Blood is protein called hemoglobin, and there are sites for binding of iron, and that causes a configuration change in hemoglobin, so that it can transport oxygen better. And this is magnesium playing a role in chlorophyll. Okay. Then we look at this idea of cooperativity. Cooperativity is, think of as friendship, friends helping friends. So binding of one molecule, one substrate in one of the sites causes the ability of the second substrate to bind much faster. So basically it's changing the enzyme, so it's going in and creating a shift in the position in the shape of the enzyme, so that the next substrates can come and bind much faster. So here we have an example of hemoglobin, which we just saw. So hemoglobin has these 4 different polypeptide chains, and the first oxygen binding causes a conformational change, so that the others are now ready to take in the other oxygens much more easily and faster. So this is a good friend. Okay, so here you have a friend coming in and making way for all the other friends to come and bind more easily. So because they are cooperating with each other to bind, for the substrates are cooperating with each other to allow each other to bind more efficiently, it's called as cooperativity. Okay, so now we are looking at the factors affecting enzyme activity. Enzymes action or activity is dependent very much on their environment. And environment is temperature, pH, enzyme concentration, substrate concentration, presence of activators, presence of inhibitors. So let's look at the temperature, pH, enzyme concentration, and substrate concentration impacts on enzymes. All of the enzymes have an optimum or optimal condition for their activity. They have the ideal condition for ideal temperature for their action, ideal pH for their option, and ideal concentrations. So let's look at heat. Here we are plotting the change in temperature to the rate of reaction, how quickly the reaction is happening. Initially, if I let's say I put enzymes and substrates in a test tube and I heat it up, the activity of the enzyme is going to increase. Why? Because as heat increases, molecules are able to move around. So heat causes movement, right? And as heat increases, molecules move much faster. As they move faster and faster, their ability to come into contact with each other is increasing. They are able to find each other because they are all agitated, they are running around. But there comes a point when the heat is way too much for their structure. And remember, in biology, structure is function. So when the heat is too high, the enzymes structure is compromised, okay, bonds start breaking. After all, bonds are also chemical energy, right? When there is too much of energy applied to it, the bonds cannot hold on to each other, they just snap. At that point, the enzyme's active site is no longer functional. The enzyme's tertiary structure is compromised, quaternary structure is compromised, and even the secondary structure is compromised. And therefore, the enzyme loses its function, its activity. So you can see the rate of reaction, let's say the product formation is going to increase for a while as the temperature increases, as the temperature is increasing. And there is a point where everything is like awesome. That's the optimum temperature. And then denaturation happens, and then it's downhill. Same thing with pH. Okay, so you have a range where the enzyme is active. But when you keep on increasing the pH, things are going to come to the point where it reaches an optimum pH, where reaction is working perfect. But when there is too much of hydrogen ions, remember pH is power of hydrogen, which means the amount of hydrogen ions in that solution. When there is too much hydrogen ions, or when there is too much hydroxy ions, then they go and interfere with the active site. If the R groups have polar molecules, and you have a lot of hydrogen ions or hydroxyl ions, they are going to go and bind to that. And therefore, your enzyme's not going to be able to bind to the substrate. So it doesn't work very well. So it goes down. See the difference in shape between temperature goes up and then goes up, as opposed to pH, where you have a more even distribution. Okay, it's a range. What about enzyme concentration and substrate concentration? So if I have, if I'm increasing, let's say I'm keeping the amount of substrate constant, okay, and I'm increasing the amount of enzymes. Remember, we don't need a lot of enzymes. We just need a few enzymes, but enough to process all the substrates. So if I keep increasing the, let's say if there is unlimited substrate, and I keep on increasing the enzymes, because there is unlimited, more and more substrate molecules are going to be modified, made into products as I'm increasing the enzymes. So that is the green line, okay, if there is unlimited substrate. But in a closed system, usually a substrate is limited, right? So if a substrate is limited, if all the substrates are being used optimally, then increasing the enzyme concentration is not going to help. So it becomes a plateau. Okay. But that's true only when substrates are limited, only when we have a small limited amount of substrates to work with. Look at substrate concentration. So here I'm keeping the same amount of enzyme, but then I'm increasing the substrates. So as I keep on increasing the substrates, as much as enzymes are available, it's going to keep on increasing. But there comes a point when the enzymes are saturated, and all the active sites of the enzymes are now being used. At this point, increasing the substrate concentration is not going to change anything. It's just going to be a plateau right here. Okay. So I'm going to look at this a little bit more. For humans, enzymes in the human body is about, our human body temperature is about 37 degrees Celsius, right? So our enzymes are also optimized to work in that range, a little bit around that range. So when you increase the temperature beyond 40 degrees Celsius, then the enzymes are denaturing, which means the protein structure is compromised, the enzyme structure is compromised, and therefore no enzyme substrate complex are forming. Same thing, if you reduce the temperature less than, this is not 400 Celsius, it's 40 degrees Celsius. Temperature is lower than 40 degrees Celsius, result in less movement. Okay, so just by looking at a graph, you can interpret it. You can say, okay, here is the optimal temperature. Too much is denaturation. Too little, it's not going to be good, because enzymes and there's not enough energy for them to move around and find each other. With pH, we saw this a little bit. It is going to, too much of hydrogen ions or too much of hydroxyl ions is going to change the properties of the active site R groups, and therefore they cannot bind to the substrate effectively. But if you have an extreme pH change, that can denature enzymes as well. And every enzyme has its own unique pH. For example, if you're looking at digestion of food, the food that you eat first goes through your mouth. You're chewing it. The saliva has an enzyme called amylase. Amylase has a pH of 7. Here, I think this is I don't have an image here, but that's okay. So pH of 7, okay. And then the food then goes down to the stomach. The stomach is a very acidic place. The pH there is about 1.8 to 2. And an enzyme called pepsin has kind of made it an optimal point to the optimal the pH of pepsin is about 2. So it can work really well in the stomach's acidic environment. And the food then travels down and goes down to the intestine, where you have an enzyme called trypsin. And trypsin works best at pH of 8, just slightly basic. So what's happening is that within the same food going from the mouth through the intestine goes through different pH conditions, where it encounters different enzymes optimized to work under those conditions. You can't have pepsin working well in your mouth. You can't have pepsin working well in your intestine. You can't have trypsin working well in the stomach. And its activity is going to be reduced at pH of 7. So every enzyme has its own specific optimum pH. This we saw, right? As enzyme increases, reaction rate increases. But when substrate is limited, then not all enzyme molecules can find the substrate. One more thing that changes the activity of enzyme is salinity. Salinity is salt concentration. So if you remember from the first unit, we looked at how sodium chloride, NaCl, is made of ionic bonds, right? Sodium gives an electron to chlorine and becomes negatively charged. Chlorine gives an electron, becomes positively charged, and then chlorine receives the electron and becomes negatively charged. And now they are oppositely charged ions, and they form a bond called as an ionic bond. But when you place sodium chloride in water, what happens is water molecules, because of their polar charges, hydrogen and oxygen having slightly positive and slightly negative charges respectively, are going to come and surround the sodium Na plus and Cl minus ions, and therefore cause the NaCl ionic bond to break between them, causing them to dissolve. And our body is essentially a aqueous solution. Aqueous system, meaning there's water. It's a water-based system. So when you have a lot of salt in a system, the changes in salinity are going to remove or add additional cations and anions, positively charged and negatively charged particles. And these can change or disrupt the attraction between amino acids, R groups. which is going to affect the secondary and tertiary structures. In other words, when you have too much of salinity or too less of salinity, then it's going to disrupt the protein or denature the protein. One of the things you see is the Dead Sea, right? Dead Sea has too much salinity, and life cannot exist. Proteins cannot perform well in that under those conditions. So here's an image just showing how temperature and pH are optimized for specific proteins. We saw pepsin, we saw trypsin. If we have amylase is going to be right here. It's going to be a little curve right here. Amylase, pH of 7. Trypsin is here and pepsin is here. Same thing with temperature. Human enzymes, 37 degrees, 37.5 degrees to 40, usually around here is optimized. But some bacteria that live in extreme conditions, like there is a procedure, there's a biotechnology tool called PCR, polymerase chain reaction. Polymerase chain reaction involves the usage of an enzyme called Taq polymerase, Thermophilus aquaticus. That's the name of the bacteria from which you get that polymerase, the enzyme polymerase. Okay, so that Taq polymerase, because it is adapted to work at really high temperatures, look at where its optimal temperature is, 72 degrees. Okay, so just to know that optimal conditions are the conditions where enzyme activity is at its best. And so we saw denaturation. Here is a nice picture. Just remember that there are 4 levels of organizations of protein, right? You have the primary structure, which is the sequence of amino acids. Then you have, so in primary structure, the amino acids are attached to each other by covalent bonds, which are strong bonds. Then you have secondary structures, which are localized hydrogen interactions. You have formation of helices called alpha helices. Then you have beta plated sheets. And then you have the R groups interacting with each other to form tertiary structures, 3-dimensional structures. And then multiple subunits in tertiary structure form are coming together to form quaternary structures sometimes. When you have changes in the conditions of temperature and pH and salinity, and what happens is that they can disrupt the R group interactions. And they can also disrupt the hydrogen bonds between the amino acids that form alpha helices and beta sheets. So in essence, you are able to break all the weaker bonds, causing the enzyme to lose its 3-dimensional shape. The primary shape still remains, but the tertiary structures and secondary structures are all lost, meaning that if there is not enough structure, if there is not a proper active site for the substrate to bind, the enzyme is useless. This is called denaturation. Under some conditions, you are able to reform the broken proteins. Not always, sometimes. When you are able to do that, and you are able to send the protein back to its original shape, that's called as renaturation. This happens, for example, here we are having beta mercaptoethanol, which is a high sulfur compound, and see what it is doing. Its addition of urea and beta mercaptoethanol is going to disrupt it, and when you are removing it, it's going to come back to its original shape. That's called renaturation. Okay, finally, we're looking at naming of enzymes. Enzymes, like, remember OSE is for carbohydrates. Typically, ASE added to, when you see ASE, it's talking about an enzyme. That's a common name. So, for example, cellulase, means an enzyme that is capable of breaking cellulose. Tyrosinase is breaking down tyrosin. Protease is breaking down protein. Lipase is breaking down lipids. Nuclease is breaking down nucleic acids. They don't describe the chemistry of the reaction, but you get a general idea of what it is about. But before this convention of adding ASE or ACE for enzymes started, there were traditional names. And they still hold on, like trypsin and pepsin. And so those are still maintained now as common names. We call them as trivial names because they were the traditional names and they don't use the ASE. So you have carbohydrates being broken down by amylase, sucrase, isomaltase, maltase, lactase, and then proteins being broken down by trypsin, peptidase, and then lipids broken down by lipase. So here are some reactions. Again, see how easy it is to understand what the enzyme is doing. Lactase, even without knowing it, you can say it is going to break down lactose. This you need to know, pepsin, trypsin, stuff like that. You need to know what it is actually doing. Kinase, you're going to see this word again and again a million times. Kinase attaches phosphate groups. So this is not as intuitive, but yeah, kinase attaches because it, think of kinetic energy, moving things forward, okay? So kinase is going to attach phosphate groups, and DNA polymerase is going to add nucleic acid bases during DNA replication. Okay, so that kind of brings us to the end of our video here. I want you to take a look at all of these questions and see if you got all the different points. How is energy moved from across systems, from the sun to chemical bonds? And what are endergonic and exergonic reactions? And what are anabolic and catabolic processes or reactions? And how are they coupled? Again, how do they go into dehydration synthesis and hydrolysis, which are commonly used in biological processes? Then we looked at energy graphs and what free energy is, and enzyme structure, how it is dependent on shape and active site, and how the activation energy is reduced by enzymes. And we looked at properties of enzymes, the ability to be recycled and reduce activation energy and so on. And then we looked at the models for enzyme action, lock and key and induced fit, competitive and non-competitive inhibition. It's the name is obvious, right? Inhibiting as competition or adding to an allosteric site. Then we looked at what can activate enzymes. We looked at cofactors versus coenzymes. And we looked at how binding of one substrate helps the binding of other substrates, which is called cooperativity. Then we looked at different conditions, environmental conditions that can affect enzyme activity, pH, temperature, substrate, enzyme concentration, salinity, etc. And denaturation and renaturation. And finally, a quick note on naming of enzymes. So hopefully this was all helpful. Thank you for listening. And if you have any questions, please let me know.